cell wall. Plant cells, in addition to the plasma membrane, have another rigid outer covering called the cell wall. What does this mean? Plant cells have the plasma membrane, but along with the plasma membrane is an outer covering, but one more outer covering is there in case of plant cells, and it is called the cell wall. So plant cells have two outer coverings. One is the plasma membrane and another is the cell wall. The cell wall lies outside the plasma membrane. So where is the position of the cell wall? It is outside the plasma membrane. The plant cell wall is mainly composed of cellulose. What is plant cell wall made up of? Plant cell wall is made up of cellulose. Now you have to remember these names. Now what is cellulose? Cellulose is a complex substance. And what is the function of cellulose? To provide structural strength to plants. So what is cellulose? Cellulose is a complex substance. It is a substance, a name given to the substance, and the function of cellulose is to provide structural strength to the plants. When a living plant cell loses water through osmosis, there is shrinkage or contraction of the contents of the cell away from the cell wall. So, very interesting. When a living plant cell loses water through osmosis, we saw osmosis, the process of osmosis in the last video. So, when plant cell loses water through osmosis, there is shrinkage. The cell shrinks, it develops wrinkles or it contracts, but it is due to the contraction of shrinkage of the contents of the cell. Whatever is there in the cell, the contents of the cell, they contract. And where are these contents of the cell placed? They are away from the cell wall. This phenomenon is known as plasmolysis. So what is plasmolysis? Shrinkage or contraction of contents of the cell. Whatever is there in the cell, those substances, they shrink, they contract, and they are away from the cell wall. Cell wall is the outer covering. So this losing of water through osmosis is called plasmolysis. We can observe this phenomenon by performing the following activity. So now let us see what is the activity. 5.6. Mount the peel of a Rio leaf in water on a slide and examine cells under the high power of a microscope. Note the small green granules called chloroplasts. They contain a green substance called chlorophyll. Put a strong solution of sugar or salt on the mounted leaf on the slide. Wait for a minute and observe under a microscope. What do we see? Now place some Rio leaves in boiling water for a few minutes. This kills the cells. Then mount one leaf on a slide and observe it under a microscope. Put a strong solution of sugar or salt on the mounted leaf on the slide. Wait for a minute and observe it again. What do we find? Did plasmolysis occur? So now here, after the activity, the main intention is to see what is plasmolysis. What do we infer from this activity? It appears that only living cells and not dead cells are able to absorb water by osmosis. So in case of, there were two cases in the activity. First, the cells were living, and the second, the cells were killed by boiling into the water. So now here, cell walls permit the cells of plants, fungi and bacteria to withstand very dilute external media without bursting. This is very important. Cell walls, the function of cell wall is to permit the cells of plants, to allow the cells of plants, fungi and bacteria. So cell wall is present in plants, fungi and bacteria. So what does the cell wall do? It allows the cells of plants, fungi and bacteria to withstand, to stand strongly in the dilute external media without bursting. The cells don't burst because of the water. The cell walls are so rigid that they help the plants, the fungi and bacteria to live properly in the dilute hypotonic external media, and they don't burst because they have the strong covering of the cell wall. In such media, the cells tend to take up water by osmosis. The cell swells, building up pressure against the cell wall. Now, what happens in such media? The cells tend to take up water by osmosis. The cells take the water inside. So the cell swells, and now pressure is created against the cell wall. The cell is inside the cell wall. The cell swells. After swelling, the cells create pressure on the cell wall, the outer covering. The wall exerts an equal pressure against the swollen cell. So the cell and the cell wall, they create pressure against each other. Because of their walls, such cells can withstand much greater changes in the surrounding medium than animal cells. So because of the cell wall, such cells can withstand, they can face much greater changes in the surrounding medium than animal cells. Animal cells don't have cell walls, so such cells in plants, fungi and bacteria, they can withstand, they can stand strongly as compared to the animal cells that don't have the cell wall. Nucleus. Remember the temporary amount of onion peel we prepared. We had put iodine solution on the peel. Why? Because why do we put iodine solution? Because iodine allows molecules to be visible under microscope. Iodine makes it the molecules to be visible easily. What would we see if we tried observing the peel without putting the iodine solution? Try it and see what the difference is. Further, when we put iodine solution on the peel, did each cell get evenly colored? According to their chemical composition, different regions of cells get colored differentially. Some regions appear darker than other regions. Apart from iodine solution, we could also use safranin solution or methylene blue solution to stain the cells. We have observed cells from an onion. Let us now observe cells from our own body. Let us take a glass slide with a drop of water on it. Using an ice cream spoon, gently scrape the inside surface of the cheek. Does any material get stuck on the spoon? So now we are taking from our mouth we are taking the inside surface, we are scraping the inside surface of our mouth under the cheek, inside surface of the cheek, so that we can observe the cells from our own body. With the help of a needle, we can transfer this material and spread it evenly on the glass slide kept ready for this. Now whatever sticky we get from the inside surface of our cheek on the ice cream spoon, we have to put it slowly with the help of a needle on the glass slide. To color the material, we can put a drop of methylene blue solution on it. Now the material is ready for observation under microscope. So why are we using methylene blue solution? To make the observation clear. Now the material is ready for observation under microscope. Do not forget to put a cover slip on it. What do we observe? What is the shape of the cells we see? Draw it on the observation sheet. Was there a darkly colored spherical or oval dot-like structure near the center of each cell? This structure is called nucleus. So again we are going after plasma membrane, after cell wall. Now we are studying nucleus. What is nucleus? When we observe the substance from the inside surface of our cheek, from our body, human body, we are observing the cells from the human body. We are now studying a new part of the cell, and that is the darkly colored spherical or oval dot-like structure near the center of each cell. This structure is called nucleus. Were there similar structures in onion peel cells? The nucleus has a double layered covering called nuclear membrane. Now we are going to discuss about the nucleus. How is the nucleus? Nucleus has double layered covering. See, two layers, two coverings called nuclear membrane. The nuclear membrane has pores, it has holes which allow the transfer of material from inside the nucleus to its outside, that is to the cytoplasm. So the area that is there outside the nucleus, it is called cytoplasm. Now the holes, the pores in the membrane, in the nuclear membrane, they allow transfer of material. Material goes in the nucleus and material comes out of the nucleus. The nucleus contains chromosomes which are visible as rod-shaped structures only when the cell is about to divide. The nucleus. Now what is nucleus made up of? Chromosomes. And what are chromosomes? Chromosomes are rod-shaped structures and they are visible only when the cell is about to divide. Chromosomes contain information for inheritance of characters from parents to next generation in the form of DNA, deoxyribonucleic acid molecules. Now chromosomes are, where are the chromosomes? Chromosomes are in the nucleus. What is the shape of the chromosomes? They are rod-shaped structures. When do we see the chromosomes? They are visible when the cell is about to divide. Chromosomes. What do chromosomes contain? They contain information for inheritance of characters from parents to next generation. So whatever features of the parents are there, they are transferred to the children, and it is possible only the features are seen in the main function of the chromosomes is to carry the characters from parents to next generation in the form of now DNA molecules. Again the new word DNA, deoxyribonucleic acid molecules. These molecules have the characters of the parents. Chromosomes are composed of DNA and protein. So we can say the rod-shaped chromosomes are made up of DNA and protein. DNA molecules contain the information necessary for constructing and organizing cells. Functional segments of DNA are called genes. Now again there are parts in DNA and they are called genes. Now see, very interesting. Chromosomes are there in the nucleus. Chromosomes have DNA and proteins, and now DNA have parts and they are called the genes. Chromosomes are composed of DNA and protein. DNA molecules contain the information necessary for constructing and organizing cells. These words you have to remember. DNA molecules. What do DNA molecules contain? DNA molecules contain the information necessary for what? For constructing and organizing cells. In a cell which is not dividing, this DNA is present as part of chromatin material. So in a cell which is not dividing, which is there as a cell, a single cell, this DNA is also present, but it is present as part of chromatin material. Now what is chromatin material is? It is visible as entangled mass of thread-like structures. It is like entangled, not so well organized. Whenever the cell is about to divide, the chromatin material gets organized into chromosomes. So first chromatin material is there, it is all entangled, it is messy, and when the cell has to divide, this chromatin material becomes organized. It becomes properly structured. It gets a proper structure like rod-like structures of chromosomes, and then chromatin material becomes chromosomes, and these chromosomes contain DNA and proteins, and these DNA contain genes, and genes are nothing but functional parts of DNA. The nucleus plays a central role in cellular reproduction. So what is the central role, important function of nucleus? It is cellular reproduction. What is cellular reproduction? What is the main role of nucleus? Cellular reproduction. But what is cellular reproduction? The process by which a single cell divides and forms two new cells. It also plays a crucial part along with the environment in determining the way the cell will develop and what form it will exhibit at maturity by directing the chemical activities of the cell. The first important function of nucleus is in the cellular reproduction. That is the process by which one single cell gets divided into two new cells. It also plays one more crucial part, and that is along with the environment in determining the way the cell will develop and what form it will exhibit at maturity. Everything is dependent on nucleus. How will it become after maturity? By directing the chemical activities of the cell. So nucleus has to look after what are the chemical activities in the cell, how the chemical activities going on in the cell, so that afterwards when the cell grows, afterwards when the cell matures, another word for maturity is growth. After the growth, how will the cell develop? In some organisms like bacteria, the nuclear region of the cell may be poorly defined. In some organisms like bacteria, the nuclear region is not properly defined due to the absence of nuclear membrane. Nuclear membrane is the covering. The covering is not there. Such an undefined nuclear region containing only nucleic acids is called a nucleoid. Now, in bacteria, what is not there? Nuclear region of the cell is poorly defined. Why? What is absent? What is not there? Nuclear membrane is not there. But such an undefined nuclear region containing only nucleic acids. So what is there? Only nucleic acids are there. And such undefined nuclear region that contains only nucleic acids is called a nucleoid. So this happens only in case of bacteria. Such organisms whose cells lack a nuclear membrane are called prokaryotes. Pro means primitive or primary. Karyote means nucleus. Now such organisms whose cells lack, the cells of these organisms do not have nuclear membrane, they are called prokaryotes. Organisms with cells having a nuclear membrane are called eukaryotes. Prokaryotic cells also lack most of the other cytoplasmic organelles. So let us see what are prokaryotes. Prokaryotes don't have other cytoplasmic organelles present in eukaryote cells. Since prokaryotes don't have nuclear membrane, so they don't have these cytoplasmic organelles. Many of the functions of such organelles are also performed by poorly organized parts of the cytoplasm. The chlorophyll in photosynthetic prokaryotic bacteria is associated with membranous vesicles, bag-like structures, but not with plastids as in eukaryotic cells. Now see the difference. The chlorophyll in photosynthetic prokaryotic bacteria don't have nuclear membrane. So chlorophyll in such bacteria is associated with membranous vesicles. Vesicles are the bag-like structures, but not with plastids. But in eukaryotic cells, what are there? There are plastids for this function. Cytoplasm. When we look at the temporary mounds of onion peel as well as human cheek cells, we can see a large region of each cell enclosed by the cell membrane. Each cell has cell membrane. This region takes up very little stain. It is called the cytoplasm. What happens? We see a large region of each cell enclosed by the cell membrane. So now each cell is having the cell membrane, but the cell membrane is closing a large region. What is this large region? This large region, other than nucleus, is called, this region takes up very little stain. It is called the cytoplasm. The cytoplasm is the fluid content inside the plasma membrane. It also contains many specialized cell organelles. Each of these organelles perform a specific function for the cell. So now cytoplasm is the fluid content. It is inside the plasma membrane. It is protected by the plasma membrane. It contains many specialized cell organelles, and these cell organelles have their own function. Cell organelles are enclosed by the membranes. In prokaryotes, beside the absence of defined nuclear region, the membrane-bound cell organelles are also absent. As we saw, prokaryotes like bacteria don't have nuclear membrane. They don't have defined nuclear region. There is absence of defined nuclear region. The membrane-bound cell organelles are also absent. Cell organelles are in the cytoplasm, but these cell organelles are also absent in prokaryotes. On the other hand, the eukaryotic cells have nuclear membrane as well as membrane enclosed organelles. The significance of membranes can be illustrated with the example of viruses. Viruses lack any membranes and hence do not show characteristics of life until they enter a living body and use its cell machinery to multiply. The significance of membranes can be illustrated with the example of viruses. Viruses, they don't have the membranes. They don't show characteristics of life. They don't show characteristics of life means unless and until they have membrane, they cannot have life until they enter a living body, and then they use its cell machinery to multiply because they don't have cell membrane.